In today's biology session, we are going to study about bacterial conjugation. Now, conjugation is the process by which one bacterium, which is called as donor cell, transfers that genetic material to another, which is called as recipient, through direct contact. Now, I want to give you an example of a businessman. Consider there is a businessman who want to give their extra money to a needful person, right? He want to do a charity. He is able to do a charity only in the case if he has extra money, right? So by donating my extra money, I became that businessman and I became the donor because I'm donating that extra money. And the person who is taking that money automatically becomes the recipient of that money, right? Just like that, the bacterium who is donating or transferring their genetic material becomes the donor, or you can call it as a F plus, and the bacteria who is receiving the genetic material becomes the recipient of it, right? Now, it can only happen if there is a direct contact between two bacteria. As I said, the donor can only donate the money, right? If they have the extra amount of money. Just like that, the donor bacterium can donate the DNA if they have extra chromosomal material in them, which is called as plasmid. Here, you can also call it as a fertility factor or F factor. Now, I'm saying this definition very confidently. Why? Because all these things are already proven. Okay, very first it was proven by Joshua and Edward in 1946. Now, as I said, it the process can happen only if there is direct contact. This was discovered by Bernard Davis in 1950 by performing an experiment in which he took a U-shaped tube. In that he filled the medium, nutrient medium, and bacteria are there. He put a filter between them, and just because the filter, the bacteria were not able to directly contact with each other, and due to that the conjugation does not occur. Then in 1952, William Hayes demonstrated that F plus strain is a donor strain, right? And the who is receiving the genetic material is a recipient of that. This donor cell contains the extra chromosomal F factor, which we call as a plasmid. It carries the gene for sex pilus formation and plasmid transfer. Now, what is this pilus formation? As I say, they need direct contact. This pilus is going to be the direct contact between these two bacterial cells. Okay, and it is used to establish the contact between F plus and F minus cell. F plus, as I said, you can consider it as a donor cell, and F minus you can consider it as a recipient cell. And the bridge or the contact between these two is called as pilus, which is going to be in donor cell, right? Now, once a contact is made between these two cells, the donor and the recipient, the F factor, which we call as a plasmid, replicates by rolling circle mechanism. What is this rolling circle mechanism? If I say it in one sentence, it is the replication of plasmid. The plasmid is going to replicate itself. Now, how this sex pillars is formed and how this rolling circle mechanism works, for that we are going to focus on this particular diagram. Okay, as you can see, this is the F plus cell, the donor cell. Why? Because they have the extra chromosomal DNA in that. And this is a recipient cell of or F minus cell. And as we know, for the transfer to be happen, they have to be physically connected, right? They are going to connect by fillers. Now, how this sex fillers is formed? Through type 4 secretion system. You can also call it as TSS. Okay. This system includes the multiple proteins which are going to be in the donor cell. Okay, it includes the cytoplasm. Okay, it is going to through a cytoplasm through cell membrane, and the cell wall is going to be enhanced in this kind of form, like just like pillars, just like bridge, and it is going to be in contact with the recipient cell, as you can see, right? This is how the sex pillars is formed by donor cell. Okay, now we have donor cell, we have a recipient cell, we have pillars between them, which is going to be useful for the transferring of genetic material. Now, if we go for rolling circle mechanism, what is going to happen here? I want you to focus here. Okay, very first, the protein complex relaxosome nicks the plasmid DNA. As you can see, it is going to come here and cut this DNA, right? Now, after that, there is going to be extent of 3 dash. Okay, as you can see, and we know the DNA transcript from 3 dash to 5 dash, right? So as you can see, the DNA is going to extend growing part, as you can see that, okay, is growing from 3 dash to 5, and due to that, because it is growing, as you can see here, it is going to be grow, and due to that, it is going to displace the parental strand, and you can see it in a red color. Okay, the 3 dash end is growing and it is displacing the parental strand, and due to that, you can see that this is the whole parental strand. Now after that, what is going to happen? As you can see, the complementary strand of that particular single stranded DNA is started forming, and this is how the whole rolling circle mechanism works. As I said, what was rolling circle mechanism? It is a replication of plasmid. Here you can see the replication occurs in the donor cell, and here you can see the one which is going to transfer in the recipient cell is also going to be double stranded. The complementary strand is forming, right? This is how the rolling circle mechanism works. Now again, I want to focus you here. I'm going to repeat this whole process. This is the donor cell, this is the recipient cell. As I said, we have the sex pillars between that. Now, what is going to happen? The protein complex of relaxosome is going to come and cut the plasmid DNA strand. After that, what is going to happen? The 3' end is going to extend, and due to that, the parental strand is going to detach from that. Okay. After that, what is going to happen? All the relaxosome proteins, as you can see here, are going to dispatch from that, but one, we can call it as an enzyme, relaxase enzyme. Okay, it is made up of protein only, so you can also call it as a, because there is a complex of protein, this relaxase is going to stay with the end of the 5' strand. Why so? Now consider that this transfer needs help of many things. Very first, it needs the help of pillars. Direct contact is needful. After that, it also needs a coupling factor. Consider this relaxase is a person who knows, he's a very intelligent person, who knows it to work, who know where to work, how to work, very intelligent, but he is blind, he can't see. Okay, consider it like that. So relaxase knows the function of itself, but they can't see, so they need to be on the location first, only then and then they can perform what they want to perform. So to show them the location, because as I say, they are blind, we are considering that this person is blind. To show them their location, there is a coupling factor. What is this coupling factor? A person who needs to help this blind person. Okay, so this coupling factor comes, hold this relaxase, and just hold this hand and take it to the location, that this is the location from where you have to enter to the recipient cell and perform your work. Right? Clear? Okay, so relaxase cannot go alone. They will perform their function and go through the sex pillars only if there is a coupling factor. So this coupling factor will come to relaxase and take it to that location. After that, what is going to happen? The DNA is going to transfer from donor cell to recipient cell. As you can see here, it is going to transfer. Okay, and as I say, the complementary strand is already started forming. When it reaches here, there is a double stranded DNA, just like the F plus cell. Okay, so in both the DNAs, the plasmid, the complementary strand itself formed by rolling circle mechanism, as we see here, right? Now, because the DNA is transferred here, the F minus, which was recipient cell, now also became the F plus cell, right? So both are F plus cell now. Now when I read the slide, you will get it. So the rolling mechanism starts where it is initiated by the complex of protein called relaxosome, which nick one of the strand of F factor at a site called OET, or you can call it as a origin of transfer, as we saw here, right? This is the OET or origin of transfer site, and the relaxosome nicks the one strand of DNA. Now what is going to happen further? Relaxase, an enzyme associated with relaxosome, remains attached with the 5' end of nick strand. As we saw here, what is going to be? The enzyme relaxase is going to attach with them. And for transferring the DNA from donor to recipient, they need a help of coupling factor. Okay, so we can see here they need the help of coupling factor to the recipient cell. Now, the frequency of recombination is very low. Why? Because chromosomal genes are rarely transferred with the independent F factor. I want you to remember this particular thing, that in normal bacterial conjugation, the frequency of recombination is low because chromosomal genes are not transferred from donor cell to recipient cell, right? And as we know, because the plasmid extrachromosomal DNA is transferred, so recipient cell also becomes the F plus cell, as we saw it in a diagram. Now, if we go for HFR conjugation, it is the type of conjugation, right? There are different types of conjugation. There is F plus type, there is HFR type. Okay, so in this particular type, the donor transfers chromosomal gene with great efficiency, but do not change the recipient bacterial in F plus cell. This is the two main difference from the normal conjugation. Very first is this transfer chromosomal gene, right? In normal conjugation, they transfer extra chromosomal gene. Here they transfer chromosomal genes also, right? And they do not change the recipient bacteria into F plus. Whether in normal conjugation we saw the recipient at the end converts themselves into F plus, right? Here it is not going to happen. Why? It is called as HFR conjugation, high frequency of recombinant produced by this mating. Now you will say, why high frequency of recombinant is going to produce? Because in normal conjugation, I told you to remember that there is a low frequency of recombination, right? We are going to know why there is a high frequency of recombination, and it is referred as HFR conjugation, and the donor is called as FFR strain. Okay, because the donor has the high frequency of recombination and they are called as HFR strain. Okay, they contain the F factor integrated into their chromosome rather than free in cytoplasm. Right, what they are going to say? That the HFR strain contain their F factor. What is F factor? The plasmid. The plasmid is integrated into the chromosome. In conjugation, we saw it was freely in the cytoplasm, right? Here, what is going to happen? Here, the F factor, which means plasmid, is integrated into the chromosome. Now, if we go for the diagram, as you can see, this is the donor chromosome. what is going to happen? The plasmid DNA is integrated, as you can see here, integrated in the normal chromosome, right? So this is the chromosome and the plasmid DNA is integrated in it. So by this process, the cell become HFR cell. Okay, how this is going to happen? As you can see, the plasmid DNA integrates in this, or it can also happen by normal conjugation if the plasmid is transferring and the pilus breaks in between, transfer breaks in between, so this HFR cell can occur. Now this HFR cell or strain becomes the F plus cell because they are going to donate their chromosomal DNA, and as this become the recipient because they are going to receive the DNA. Now as you can see, the pilus is formed between them. Now if you have understand the conjugation very well, now you will know how this pilus form by type 4 system, right? After that, what is going to happen? There is going to perform a, as you can see here, the rolling circle mechanism in which relaxosome is going to work, relaxase is going to work. The process is going to just like conjugation. Okay, but there is a difference. I will tell when there is a difference from conjugation. The very first difference is this, that there is plasmid, but it is not freely available in the cytoplasm. It is integrated in the chromosomal DNA. Now as you can see, the pilus is formed between them. After that, what is going to happen? The rolling circle mechanism is going to start, just like the one we saw it in a conjugation, right? The relaxosome protein will come, then it will grow the 3' end, and the parent's strand is going to be displaced and going to transfer from donor to the recipient DNA. Now what is going to happen? The transfer is going to be break in between. This pilus is going to be break in between the transformation. Why this happened? Because the plasmid DNA was a small DNA. If you remember, I said small extra chromosomal DNA. But here the whole chromosomal DNA is going to be transferred. Right, so this is a large amount of genetic material which is going to take time. If you want to transfer the whole DNA, it is going to take time. For example, for E. coli, it took 100 minutes to transfer the whole DNA, right? Now, because this is a process, at some point, this pilus is going to break. That happens in that the pillars break in between, and this is the reason that not the whole DNA but only the part of the DNA is transferred from the F plus cell to F minus cell, as you can see here, right? Now what is going to happen? They are going to form their complementary strand. Here they form their complementary strand. Here also it is going to form the complementary strand, but it is incomplete, as you can see, incomplete plasmid. This is the reason it is going to be F minus only. It is not going to change in F plus, as we saw in the conjugation. In the end, both cells become F plus, right? Here there are two possibilities. Very first possibility is that this incomplete plasmid will degrade itself, and then there is a no use of it. In that case also F minus remains F minus, right? Because there is no transfer of DNA, because the transfer of DNA which was happen is degraded. Now there is a second possibility, okay, which is a great possibility, that the incomplete DNA will integrate in the chromosomal DNA, as you can see here, right? The incomplete DNA will integrate in the chromosomal DNA, and this also remains the F minus. Why? Because there is not whole DNA which is transferred here. Remember, only the part of DNA is transferred here. Now this particular cell is called as recombinant cell. Why? Because there is a recombination of that chromosomal DNA, as you can see. Now this cell is very much helpful because especially in recombination, we say high frequency recombination, right? Why? Because when this cell is going to replicate again and again, there is going to be chance that this part, this incomplete part, is again going to detach from the chromosome. Now when the part is going to detach, some part of chromosomal DNA also detached from it, right? Whenever it is going to detach, some part of chromosomal DNA is also going to detach and form a extra chromosomal material here, just like here, form a plasmid here. Now after they form a plasmid, because of that, that plasmid can then transfer by different genetic transfer methods, and due to that the chances of recombination becomes more. So this is how the HFR conjugation is performed. Now if I'll go from slide, okay, we saw that there is a high frequency of recombinant, right? DNA transfer begins when the integrated F factor is nicked as its origin transfer site. As we saw, how it is going to nick? Just like we saw it in a conjugation system. The protein complex reloxosome is going to come and nick one of the strand from that origin of transfer site. As it is replicated, how it is going to be replicated? By rolling circle mechanism, right? The chromosome moves to recipient. Now here, as I say, only the part of F factor or plasmid is initially transferred and the host chromosome follows it. Okay, transfer of entire chromosome with integrated F factor requires about 100 minutes in E. coli. As I give an example, it took 100 minutes in E. coli if you want to transfer the entire chromosome. Okay, but connection between the cell usually break before this process is finished, right? As I say in the diagram, that they does not give you the chance of transferring the whole genetic material, right? Because it is too large for it. So it breaks the connection in between. Thus, a complete F factor is rarely transferred, and that's why the recipient cell remain F minus. As I say, there are two chances. One, that the DNA which is transferred from donor to recipient is going to degrade, or second, there is a possibility that it became recombinant cell. After the replicated donor chromosome enters the recipient cell, and it may degrade, as I say, or incorporated into F minus genome by recombination. So this was the whole conjugation and HFR conjugation. I hope you got the point of conjugation and how the two bacteria connect with each other and transfer their DNA. That is just like as I give an example of that businessman who needs to charity. Only they can charity if they have the extra money. Just like that, the bacteria can donate only if they have extra chromosomal material. They need direct contact for that and the rolling circle mechanism. And this is how they transfer their extra chromosomal DNA or chromosomal DNA in case of HFR from donor cell to recipient cell. I hope you got the point. Thank you.